more than a kilometer below the surface, in water that stays near 4 degrees Celsius year-round, where no sunlight reaches and no season has ever been felt. Something has been waiting for 80 million years, not hiding, not struggling, just existing, unchanged, unhurried, in the same cold dark it occupied before the asteroid that killed the dinosaurs, before the continents settled into the shapes we recognize today, before almost everything alive on this planet had yet appeared. The frilled shark is one of the oldest predator designs still operating on Earth. Its teeth, 300 of them, arranged in 25 inward curving rows, are not sharp in the way a knife is sharp. They are sharp in the way a trap is designed. Anything that enters that mouth and moves in the wrong direction only goes deeper, and yet no human being has ever seen it use them. Every specimen ever studied was already dying by the time anyone reached it, pulled from its world into pressure and light it was never built to survive. Everything we know about this animal comes from creatures already removed from the only place they have ever known. The frilled shark hunting in the dark, doing the thing it was made for, has never been witnessed by human eyes. An animal this old, this unchanged, this completely adapted to a world we can barely reach, and we still do not fully know what it does down there. Tonight, we go as far into its world as the evidence allows. Before we go further, if this is the kind of content you come back for, subscribe and leave a like. It helps more than you'd think. Now let's descend into the part of the ocean that time forgot to change. There is a tendency to imagine ancient life as something buried, sealed beneath layers of stone and distance, belonging to a world that ended long ago. Yet the story of the frilled shark begins with a different kind of age, one measured not by extinction, but by continuity. 80 million years ago, the Atlantic Ocean was narrower than it is today. The landmass that would eventually become India was still moving northward across an ancient sea, not yet joined to Asia, not yet lifting the vast wall of mountains that would become the Himalayas. On land, the most advanced primates were small mammals moving through branches and leaves, creatures so distant from humanity that they scarcely resemble the idea of a human ancestor at all. Even then, the lineage that would produce the frilled shark was already established. Yet even that span reaches back only partway through the larger story. Sharks themselves are among the oldest surviving vertebrate lineages on Earth. Their origins stretch back roughly 450 million years before forests spread across continents, before vertebrate animals walked on land, before many of the ecosystems familiar today had even begun to exist. The world has reinvented itself repeatedly during that interval, continents assembling and breaking apart, climates warming and cooling, entire dynasties of life appearing and disappearing. Through all of it, sharks remained. The frilled shark occupies a particularly ancient branch of that family tree. It belongs to a group known as hexanforms, a lineage that separated from other sharks near the base of shark evolution itself, long before the sleek forms of modern ocean predators emerged, long before the refinements that produce the familiar silhouettes of great white sharks, hammerheads, and tiger sharks. This branch had already begun following its own path. What makes this remarkable is not merely age, but isolation. The surface ocean is a place of continual disruption. Storms reshape habitats. Seasons alter temperatures and food availability. Sunlight drives cycles of abundance and scarcity. Across millions of years, these shifting conditions create new challenges, and evolution responds by producing new solutions. Far below, the deep ocean operates according to different rhythms. At depth, temperatures can remain astonishingly stable, varying by less than 2 degrees Celsius, or about 36 degrees Fahrenheit, across spans of time that exceed recorded human history many times over. There are no seasons, no hurricanes, no sunlight reorganizing entire food webs. The environment changes so slowly that the pressure to adapt can become almost imperceptible. Evolution does not create novelty simply because time passes. It responds to problems. When conditions shift, organisms must shift with them. When conditions remain stable, successful designs can persist for extraordinary lengths of time. The frilled shark entered such a world. Its scientific name reflects the impression it made upon the people who first examined it. Chlamydoslacus anguineus combines words rooted in ancient languages. The first refers to a mantle, inspired by the unusual frilled structures surrounding its gills. The second means snake-like, a reference to the elongated body that seemed unfamiliar even to experienced zoologists. The name reads almost like a record of scientific uncertainty, an attempt to describe something that appeared to belong to several categories at once, and none of them comfortably. That uncertainty has never entirely disappeared. For more than a century, Chlamydococcus anguineus was considered the sole living representative of its kind. Then, in 2009, researchers formally described a second species, Chlamydococcus africana, from waters near southern Africa. The discovery revealed that this lineage still held secrets in regions already being studied, suggesting that its true story remained incomplete. Everything that follows grows from this foundation. Every unusual structure, every unanswered question, every difficulty involved in studying or protecting this animal traces back to one central fact. The frilled shark found an environment that asked almost nothing new of it, across immense stretches of time, while the surface world continued to transform, remaining exactly what it was proved sufficient. Imagine laying the outlines of every familiar shark on transparent sheets of glass and stacking them one atop another. The shapes would begin to converge. Broad pectoral fins would stretch outward like wings. Powerful tails would narrow into efficient crescents. Jaws would sit beneath elongated snouts, tucked into positions refined by immense spans of evolutionary trial and adjustment. Across oceans and across time, many shark lineages arrived at remarkably similar solutions. The frilled shark sits outside that pattern. It carries a body plan that seems to have stepped away from the main current of shark evolution long before the modern ocean took shape. While other sharks gradually transformed into streamlined pursuit predators, this lineage retained a form that feels older, closer to the deep foundations from which sharks first emerged. Its mouth is the first indication. In most living sharks, the jaw rests beneath the head, angled downward and slightly back from the tip of the snout. This arrangement improves hydrodynamics and supports rapid forward swimming. The frilled shark follows a different blueprint. Its jaw occupies the very front of the face, facing directly ahead. This position is considered ancestral among vertebrates, a reminder of anatomical arrangements that existed long before many later refinements appeared. Looking at the animal from the side, the mouth does not seem tucked beneath the skull. It appears to lead the way. The rest of the body follows a similarly unusual logic. A single dorsal fin rises near the rear portion of the animal, positioned much closer to the tail than in most sharks. In faster species, dorsal fins function as stabilizing structures, helping control rolling movements during active swimming. Here, the fin's placement suggests that high speed was never the central demand shaping the body. Stability remains important, but it is achieved through a different balance of form and movement. Even details hidden from casual observation reveal the same pattern. Male frilled sharks lack an anal fin, a feature commonly present in many other shark groups. The exact consequences of this difference remain uncertain. It persists as one of several anatomical details that remind researchers how far this lineage stands from the standard shark template. The pectoral fins offer another clue. Rather than broad lifting surfaces designed to generate significant hydrodynamic support, they are relatively small and rounded. They function more like stabilizing paddles than aerodynamic wings. Their role appears less concerned with rapid maneuvering and more concerned with maintaining position and balance within the water column. Between these fins stretches a remarkably elongated trunk. The proportions feel almost inverted when compared with many familiar sharks. Fin surface area occupies a modest fraction of the body's overall form, while the torso extends in a long, uninterrupted curve. The result is an animal that appears composed more of body than of appendage, more flowing tube than streamlined projectile. This shape changes how movement is distributed. Instead of concentrating propulsion primarily in the tail, the frilled shark can involve much of its body in locomotion. Flexibility becomes a structural asset. Motion travels through the trunk in gradual waves, carrying the animal forward through water with an economy suited to a world where wasted energy carries consequences. Seen together, these features form something more meaningful than a collection of oddities. They represent an alternative answer to a problem that countless shark lineages confronted across geological time. Most evolved toward greater speed, greater streamlining, and increasingly specialized forms of pursuit. The frilled shark remained on an older path, preserving a configuration that many of its distant relatives gradually left behind. What emerges from that persistence is not a failed experiment, nor an unfinished design. It is a surviving example of a possibility that once existed more broadly within shark evolution. The familiar shark silhouette came to dominate the oceans, yet far below, one ancient branch continued carrying a different inheritance, proving across 80 million years that another solution remained viable all along. Along the throat of the frilled shark runs a detail so unusual that it often captures attention before any other feature. It is not the shape of the body, nor the arrangement of the fins, nor even the remarkable mouth. Instead, it is a series of folds that seem almost decorative at first glance, delicate ridges draped across the neck like fabric gathered by an unseen hand. These folds mark the locations of the animal's gill slits. Among living sharks, 5 gill slits became the prevailing arrangement long ago. Across countless species inhabiting reefs, coastlines, open oceans, and abyssal waters, this pattern repeats with remarkable consistency. It is one of the defining characteristics people unconsciously recognize when they picture a shark. The design became so widespread that it appears almost inevitable. The frilled shark follows an older pattern. Instead of 5 gill slits, it possesses 6. Each slit remains fully functional, participating in the constant exchange that sustains life beneath the sea. More striking still is the tissue surrounding them. The edges are not smooth, they are ruffled and folded, extending outward in soft fringes that overlap with neighboring fringes. Together, they create the appearance of a continuous frill, encircling the throat, as though the boundary between one gill opening and the next has been partially erased. This unusual structure inspired both the animal's common name and its scientific identity. It is also the feature most frequently mentioned by researchers encountering the species for the first time. Before measurements are taken or anatomical comparisons begin, the eye is drawn almost immediately to those layered folds. They create the impression of encountering something carried forward from a much earlier chapter of vertebrate history. Yet the frill is not merely ornamental. Its shape serves a practical purpose. Every fold increases the total surface area available for respiration. The principle is simple. Oxygen enters the bloodstream through delicate respiratory tissues. The greater the area exposed to flowing water, the greater the opportunity for oxygen molecules to cross into the body. By expanding the effective surface through elaborate folding, the frilled shark extracts more oxygen from each volume of water passing across its gills. This becomes particularly valuable for an animal whose lifestyle differs from that of many familiar sharks. Numerous shark species rely heavily on continuous forward movement. As they swim, water is driven across their respiratory surfaces, ensuring a steady supply of oxygen. Their bodies are built around motion, and breathing is intimately linked to that motion. The frilled shark appears to operate under a different set of priorities. Its respiratory architecture suggests efficiency at lower flow rates, allowing oxygen extraction even when water moves gradually across the gills. In such a design, patience becomes compatible with survival. The body does not demand constant movement to satisfy its respiratory needs. Water drifting across the throat can be used effectively, each fold contributing to a system refined not for speed, but for economy. This six-gill arrangement is not unique to the frilled shark alone. Other members of the ancient order Hexantiformes retain similar characteristics. The six-gill and seven-gill cow sharks share this departure from the standard five-slit pattern, linking them through a common evolutionary inheritance. The trait functions almost like a family signature, preserved across lineages that separated from other sharks deep in geological time. Fossils reveal that sharks bearing six gills existed hundreds of millions of years ago. The feature appears again and again in ancient deposits, tracing a lineage that stretches backward far beyond the rise of mammals and deep into eras when entirely different forms of life dominated the planet. For many shark groups, evolutionary history revised that arrangement. Additional refinements emerged, body plans shifted, and older traits gradually disappeared. The frilled shark retained this one. Each exchange of oxygen through those six openings follows a pattern inherited across immense spans of time, a rhythm carried forward from ancestors already swimming through ancient seas, while the first flowering plants were only beginning their spread across the earth. 300. Before shape, before function, before biology, there is simply the number itself. 300 teeth contained within a mouth not much wider than a large human hand. It is a density that feels almost excessive, as though nature continued adding points long after necessity had been satisfied. Yet every one of them has a purpose. In many sharks, teeth exist in a hierarchy. A primary row performs most of the work, while replacement rows wait behind, advancing only when older teeth are lost. The arrangement resembles a reserve force held in readiness. The frilled shark follows a different strategy. Its teeth are organized into roughly 25 rows distributed across the upper and lower jaws, and these rows exist together as a functional structure rather than a simple queue of replacements. Looking into the mouth reveals not a single line of weapons, but a dense field of them, layered one behind another in a pattern that appears almost architectural. The shape of each tooth deepens the impression. Rather than ending in a single point, every tooth divides into three slender cusps rising from a common base. Viewed head-on, the profile resembles a trident carved in miniature. The central cusp extends slightly farther than the two flanking points, creating a crown of three sharpened projections. Repeated hundreds of times, the pattern transforms the interior of the mouth into a forest of delicate hooks. The crucial detail lies in their orientation. None of these points aim outward. None project sideways. Every cusp bends inward, toward the throat. The curvature remains remarkably consistent throughout the jaw. Tooth after tooth follows the same geometry, forming a directional system rather than a collection of independent structures. Once something enters that system, the consequences become mechanical. Imagine the body of a squid passing between those rows. The initial contact may involve only a few teeth, yet any movement away from the throat immediately changes the interaction. The inward curve causes the points to engage more firmly. Retreat increases contact. Resistance deepens engagement. The prey's own motion contributes to its restraint. A fish attempting to reverse direction encounters the same problem. The geometry converts escape into pressure and pressure into capture. With every backward movement, the curved cusps gain a stronger hold. The design does not depend upon crushing force or repeated tearing motions. It functions because the structure itself guides movement in only one useful direction. This is why the teeth are best understood not as cutting tools, but as a retention system. They form a biological one-way valve. Once prey crosses a certain threshold, the architecture of the mouth begins assisting the predator. The trap operates through shape more than strength. Time, however, wears down even the most effective structures. Teeth break, points become dulled, contact with prey gradually erodes the sharpest edges. The frilled shark answers this problem through continual renewal. Like other sharks, it is capable of replacing teeth throughout its life. New teeth develop within the jaw and gradually move forward as older teeth wear away. The process never truly ends. The arrangement renews itself continuously, maintaining the effectiveness of the entire system across decades. It is less like a set of tools and more like a living mechanism that repairs itself while remaining in operation. What makes this especially remarkable is how little the design has changed. Fossil teeth attributed to frilled shark relatives display an astonishing resemblance to those found in modern specimens. Across immense stretches of geological time, the same three-pointed structure appears again and again. The proportions remain familiar. The curvature remains familiar. The overall geometry persists with extraordinary fidelity. Many anatomical features drift and transform as environments change and new pressures emerge. These teeth seem to have encountered no such demand. Their design arrived at a solution and retained it. Seen in that light, the mouth becomes something more than a collection of sharp structures. It is evidence of a strategy refined long ago and preserved because it continued working. The frilled shark does not require exceptional speed, overwhelming power, or relentless pursuit. It requires only proximity, timing, and the brief closing of a jaw built around 300 inward turning points, each contributing to a mechanism that has endured for millions upon millions of years. Movement leaves signatures long after the movement itself has vanished. A track pressed into mud, a ripple spreading across water, a branch bending back into place after something passes through. In biology, entire theories can emerge from such traces. The frilled shark has offered science little more than traces. What can be observed directly is the shape of the body in motion. Unlike many familiar sharks that generate thrust primarily through powerful sweeps of the tail, the frilled shark distributes movement across nearly its entire length. Waves travel from head to tail in a flowing lateral pattern, bending the trunk from side to side. The motion resembles that of eels, sea snakes, and several ancient fish lineages whose bodies rely on flexibility rather than concentrated bursts of power. There is an elegance to this arrangement. Instead of pushing large volumes of water aside through force, the animal advances through a sequence of coordinated curves. Each bend contributes a small amount of forward momentum, The result is slower than the rapid propulsion used by many pelagic predators, yet it is also economical. Energy is spent carefully. In environments where food may arrive unpredictably and long intervals separate successful meals, efficiency becomes a valuable form of survival. The body itself appears built around this principle. The trunk extends for much of the animal's length, while the fins occupy relatively modest proportions of the overall profile. Cartilage provides flexibility throughout the skeleton, allowing curves to travel smoothly from one region of the body to another. Rather than resisting bending, the structure seems designed to accommodate it. Undulation becomes not merely possible, but natural. Even the placement of the dorsal fin contributes to this impression. Positioned unusually far toward the rear, close to the tail, it leaves most of the body unobstructed. The forward portion remains free of large, stabilizing structures that might interfere with rapid extension. Seen through the lens of movement alone, the arrangement suggests an animal whose most important actions occur at the front, while much of the supporting machinery remains behind. From these observations emerges one of the most intriguing ideas in frilled shark biology. Several researchers have proposed a hypothesis based on anatomy rather than direct observation. The idea remains unconfirmed, yet it continues to attract attention because it aligns so neatly with the available evidence. According to this interpretation, the frilled shark may draw part of its body into a curved configuration before attacking. The posterior region could function somewhat like a compressed spring, storing mechanical energy within a series of muscular bends. When released, that stored energy might propel the front of the body forward in a sudden lunge. The comparison most often invoked is a striking snake. A predator appearing slow during ordinary movement can become unexpectedly rapid during the final instant of an attack. The contrast between apparent pace and actual striking distance becomes part of the deception. An approaching prey animal may judge danger based on observed swimming speed, only to encounter a burst of motion that exceeds those expectations. Calculations based on flexibility and body proportions suggest such a strike could extend farther than the silhouette alone would imply. A long trunk provides room for stored curvature. A flexible skeleton permits rapid straightening. A forward-facing jaw benefits from arriving directly toward the target rather than from beneath it. Yet every part of this remains an inference. No camera has documented a frilled shark coiling its body before an attack. No recording has captured the instant of a strike. No human observer has witnessed the complete sequence unfold in the animal's natural environment. The hypothesis survives because anatomy points toward it and because stomach contents demonstrate that prey is being captured somehow. Between those two facts lies a space filled largely by deduction. This creates an unusual situation. The behavior most closely tied to survival remains among the least understood aspects of the species. Structures can be measured, skeletons can be examined, muscles can be mapped. The action those structures were built to perform continues to reside beyond direct observation. And so, somewhere beyond human sight, a body may bend into a curve that no camera has recorded. Muscles may gather tension through movements perfected across immense spans of time. Then, in a brief instant, hidden within the larger darkness, the curve may unfold, completing a sequence known only through its consequences. A closed frilled shark reveals surprisingly little. Seen from the outside, the head appears narrow, almost restrained. Nothing in its resting profile immediately suggests the scale of what it can consume. The most important parts of the feeding apparatus remain folded away, hidden within cartilage and muscle, waiting for a moment that has almost never been witnessed directly. The arrangement begins with the position of the mouth itself. In most sharks, the jaws sit beneath the snout. When feeding, the animal must angle its body and bring the mouth upward toward the target. The strike becomes a coordinated movement involving both the jaws and the trajectory of the swimmer. The frilled shark follows a different geometry. Its mouth occupies the very front of the head, facing directly ahead. Prey located in front of the animal does not require substantial repositioning. The feeding apparatus is already aimed toward the destination. This seemingly simple difference alters the entire mechanics of capture. The jaws do not merely open, they extend. When feeding, the frilled shark can project its jaws forward from the skull, increasing the distance between the teeth and the rest of the head. The effect is subtle when viewed through anatomy diagrams, yet significant in practice. The reach of the strike becomes greater than the outline of the closed mouth suggests. A target judged to be beyond immediate range may suddenly find that the range has changed. Cartilage plays an important role in this transformation. The structures connecting the jaws to the skull are comparatively flexible, allowing a degree of mobility uncommon among many predatory fishes. Rigidity often contributes to stronger biting forces, but flexibility offers different advantages. It permits expansion, extension, and a wider opening angle. The mouth can therefore achieve an impressive gape relative to body size. The upper and lower jaws separate dramatically, creating an entrance capable of accommodating prey much larger than one might expect from the shark's proportions. Rather than relying upon crushing power, the system prioritizes reach and volume. Evidence of this strategy appears repeatedly in stomach examinations. Researchers studying specimens recovered from fishing operations have discovered entire squid within the digestive tract, some representing a substantial fraction of the shark's own length. These prey items were not torn into small fragments before swallowing. In many cases, they were consumed largely intact, suggesting a feeding method designed to engulf rather than dismantle. Among the most commonly recovered remains are squid beaks. These hard structures resist digestion long after softer tissues disappear, leaving durable clues behind. Their frequent presence indicates that squid form a major component of the frilled shark's diet. This presents an intriguing contrast. Squid are often agile animals. They propel themselves through the water by expelling jets of water from the mantle, accelerating rapidly when threatened. Their reactions can be swift and unpredictable, yet they repeatedly appear within the stomachs of a predator whose outward appearance suggests patience rather than speed. The menu extends beyond squid. Researchers have also documented bony fishes and fragments of other sharks. Such findings portray an opportunist rather than a specialist. The frilled shark appears willing to exploit whatever suitable prey becomes available, adapting its feeding choices to the opportunities offered by its environment. Viewed together, the anatomy begins to form a coherent picture. A forward-facing mouth eliminates the need for complex body alignment. Protrusible jaws increase reach. A wide gape accommodates unexpectedly large prey. Rows of inward curving teeth ensure that whatever crosses the threshold remains moving in only one useful direction. The strategy depends less upon overpowering prey than upon exploiting a brief moment of proximity. Success may hinge on approaching undetected, extending farther than expected, and transforming a small miscalculation into a final one. Yet even after examining the jaws, the stomach contents, and the remarkable architecture of the mouth, an important uncertainty remains. The prey most commonly recovered from frilled sharks often possesses speed, maneuverability, and strong escape capabilities. The anatomy explains what happens after contact occurs. The deeper mystery lies in the moments beforehand. Somewhere between detection and capture, a gap still exists in the story. The frilled shark crosses that gap routinely enough to survive, but the precise manner in which it does so remains hidden within a behavior that science has not yet observed directly. Imagine descending through the ocean not as a diver, but as a measurement. With every meter traveled downward, conditions shift according to rules that cannot be negotiated. Light weakens, pressure increases, temperature falls. The world becomes less a landscape and more a set of physical realities layered one beneath another. The frilled shark spends its life within one of those layers. Confirmed specimens have been recovered from depths ranging roughly between 200 meters, which is about 124 miles, and 1,500 meters, which is about 932 miles. Most encounters, however, occur between 500 meters, which is about 311 miles, and 1,000 meters, which is about 621 miles. These depths occupy a region where the character of the ocean changes profoundly. Near 200 meters, which is about 124 miles, the last usable sunlight disappears. Above this boundary, enough light remains to support photosynthesis. Microscopic organisms convert sunlight into biological energy, forming the foundation of vast marine food webs. Below it, that process ceases. Energy can no longer be manufactured directly from sunlight. Life must instead depend upon resources arriving from elsewhere. This transition reshapes the entire economy of existence. Farther down, around 500 meters, which is about 311 miles, pressure reaches approximately 50 atmospheres. Every portion of the frilled shark's body experiences forces that would rapidly overwhelm organisms adapted to surface conditions. Yet for the shark, these forces are not extraordinary. They are ordinary. Its tissues, organs, and fluids developed within this environment and operate as though such pressure were simply the natural state of reality. Temperature follows a similar pattern of stability. Across much of the frilled shark's range, water temperatures remain between 2 degrees Celsius, or 36 degrees Fahrenheit, and 7 degrees Celsius, or 45 degrees Fahrenheit. Unlike surface environments, these conditions do not fluctuate dramatically with the seasons. There is no annual cycle of warming and cooling, no summer abundance followed by winter scarcity. The temperature remains remarkably consistent year after year, century after century. That consistency shapes everything. Without strong seasonal rhythms, food does not arrive according to a dependable calendar. Surface oceans experience blooms of productivity tied to sunlight and climate. Deep water receives no such regular schedule. Nutrients drift downward irregularly. Organic material arrives in varying amounts. Long periods of scarcity can separate opportunities for feeding. Survival favors animals capable of enduring uncertainty without excessive energy expenditure. Yet this world is not completely without rhythm. Twice each day, one of the largest biological movements on Earth passes through the depths inhabited by the frilled shark. Vast populations of mesopelagic organisms undertake a vertical migration between deeper and shallower waters. Lanternfish, small squid, crustaceans, and countless other creatures rise toward the surface during darkness and descend again with the return of daylight. The scale of this migration is immense. Trillions of animals participate across the global ocean. Collectively, they form a drifting layer of life, moving upward and downward through the water column every 24 hours. For predators living at depth, This movement represents something rare: predictability. Prey that may otherwise be scattered across enormous volumes of ocean passes through familiar depth ranges with remarkable regularity. The migration creates recurring opportunities within an environment where certainty is uncommon. Everything about the frilled shark appears aligned with these conditions. It is not constructed for speed over long distances. It is not built for maintaining a high metabolic pace. Its biology reflects patience, conservation, and efficiency. The surrounding environment rewards exactly those qualities. In this sense, the deep ocean is more than a habitat. It is a partnership between physics and anatomy. Pressure, temperature, energy availability, and biological cycles fit together with the shark's design so completely that separating one from the other becomes difficult. The animal belongs to these conditions not through preference, but through correspondence. Over immense spans of time, its body and this layer of the ocean have shaped one another into a remarkably precise match. Close your eyes and imagine identifying a moving object without seeing it. Not by sound, not by touch, not by any sense familiar enough to describe easily. The challenge begins to approach the world inhabited by the frilled shark, a world where vision remains useful but no longer occupies the central role it does for creatures of the surface. At depths around 500 meters, which is about 311 miles, ambient light has diminished to the point that image-forming vision becomes severely constrained. Shapes fade. Distances lose clarity. The ocean ceases to be a place where sight alone can reliably explain what surrounds an animal. The frilled shark answers this problem through an array of sensory systems that transform water itself into information. Running along the length of the body and extending across the head is the lateral line system, a network of fluid-filled canals connected to highly sensitive receptors. These structures respond to tiny disturbances moving through the surrounding water. A swimming fish displaces water. A drifting squid alters local pressure patterns. Even subtle movements create waves that spread outward through the environment. The shark does not need direct contact to perceive them. Instead, the lateral line functions as a kind of distant touch, allowing the animal to detect motion through the distortions it leaves behind. Pressure changes become signals. Water movement becomes evidence. The environment continuously writes information into the surrounding fluid, and the lateral line reads it. In the frilled shark, this system appears especially well developed relative to body size. Such investment makes sense for an animal that cannot depend entirely on vision. Detecting movement may matter more than identifying precise shapes. The difference between empty water and the passage of prey can exist within a disturbance too subtle for human senses to notice. Yet pressure is only one language spoken by living organisms. Every muscle contraction generates tiny electrical currents. Every nerve impulse contributes to a faint bioelectric field surrounding the body. Across the head of the frilled shark are clusters of specialized sensory organs, known as the ampullae of Lorenzini. These pore-like structures detect electrical activity within the water, granting access to another layer of information invisible to ordinary sight. A squid accelerating through the ocean provides an example. The contraction of its mantle muscles creates electrical signals as part of normal biological function. Those signals leak into the surrounding environment. The exact range at which the frilled shark can detect them remains uncertain, but researchers understand that the sensitivity is meaningful enough to assist in locating prey under conditions where visual information is limited. The arrangement of these organs is revealing. The snout is relatively short and rounded, concentrating many of the electroreceptive structures into a forward-facing cluster. Their distribution aligns neatly with a predator whose attention focuses on what lies directly ahead. Chemical information adds yet another dimension. Deep sea sharks possess substantial olfactory capabilities, and evidence suggests that the brain regions devoted to processing scent are proportionally enlarged. In cold, dense water, dissolved chemical traces behave differently than they do near the surface. Odors may persist longer and travel through the environment in ways that create navigable gradients. A predator can potentially follow these gradients, much as a traveler follows a distant path. Even vision retains an important role. Behind the retina lies a reflective structure known as the tapetum lucidum. This layer redirects incoming light back through the visual receptors, increasing the likelihood that scarce photons will be detected. Rather than wasting faint illumination, the eye effectively grants itself a second opportunity to capture it. And there is light to capture. An estimated 76% of deep-sea organisms produce their own illumination through bioluminescence. Tiny flashes, drifting glows, pulses, streaks, and brief sparks emerge from living bodies throughout the water column. The darkness is not empty. It is patterned with biological light generated by the organisms themselves. Taken together, these senses create a perception difficult to translate into human terms. The frilled shark experiences pressure gradients, electrical fields, chemical traces, and scattered points of living light simultaneously. Its environment arrives not as a simple visual scene, but as overlapping layers of information, woven together into a picture of the world that exists far beyond the limits of ordinary sight. Long before an animal can hunt, sense, or move through its environment, it must solve a simpler problem. It must remain where it wants to be. In water, gravity never disappears. Every creature is pulled downward continuously, and every creature must develop some response to that pull. Many bony fish answer the problem with an internal chamber known as a swim bladder. Filled with gas and adjusted according to depth, this structure allows the animal to fine-tune its buoyancy with remarkable precision. By expanding or contracting the volume of gas inside the body, a fish can rise, sink, or remain suspended with minimal effort. Sharks inherited a different path. As cartilaginous fish, they never evolved a swim bladder. Instead, they developed alternative solutions built around body composition and internal chemistry. Among sharks, the frilled shark represents one of the most extreme examples of this strategy, relying heavily on an organ that at first seems unrelated to floating at all. The organ is the liver. In most vertebrates, the liver performs a variety of metabolic functions, processing nutrients and supporting the chemistry of life. In the frilled shark, it assumes an additional responsibility. It becomes a buoyancy device. This is possible because the liver is saturated with squalene, a hydrocarbon lipid whose density is lower than that of seawater. While seawater possesses a specific gravity of approximately 1.025, squalene possesses a specific gravity of approximately 0.86. That difference may appear small on paper, yet across a large enough volume, it creates meaningful lift. The frilled shark possesses precisely such a volume. Its liver is enormous relative to the rest of the body. In some specimens, the organ accounts for as much as 25% of total body mass. This is not a modest anatomical feature occupying a corner of the abdominal cavity. It is a major structural component of the animal itself, representing one of the highest liver-to-body mass ratios recorded among shark species. A quarter of the body devoted to a single organ reflects a profound evolutionary commitment. The result is not perfect neutral buoyancy, The frilled shark still makes adjustments through movement and posture. Water is dynamic, and exact balance requires continual refinement. Yet the liver dramatically reduces the amount of active swimming necessary to remain within a preferred depth range. Instead of constantly generating upward thrust to counter sinking, the animal receives passive assistance from its own internal chemistry. The same organ performs a second role. Food is unpredictable in the deep ocean. Days may pass without opportunity. In some cases, intervals may extend even longer. Under such conditions, stored energy becomes invaluable. Squalene is energy-rich, allowing the liver to function as a reserve of metabolic fuel. The organ that helps the shark float also serves as a biological battery, preserving calories for periods when prey is absent. The dual function already makes the liver remarkable, yet there is evidence that a third contribution may be equally important. Mass distributed within a body influences how that body naturally settles in water. The location of the liver relative to the center of buoyancy affects balance, orientation, and trim. Subtle shifts in weight distribution can influence the angle at which an animal rests or drifts. Some researchers have suggested that the frilled shark's unusually large liver may contribute to the posture it adopts while wading within the water column, though the precise effects remain incompletely understood. What emerges is a solution characterized by consolidation rather than specialization. Instead of evolving separate structures for buoyancy, energy storage, and aspects of body balance, the frilled shark channels multiple needs into a single anatomical investment. Within the conditions of the deep ocean, this arrangement makes profound sense. Speed is rarely essential. Efficiency is. Every calorie preserved matters. Every unnecessary movement carries a cost. A large liver filled with low-density energy-rich compounds addresses several challenges simultaneously. The organ becomes flotation device, fuel reserve, and stabilizing influence at once. What appears unusual at first glance reveals itself as a remarkably elegant compression of function, refined across immense spans of time until complexity settled into something deceptively simple. Among land animals, the African elephant has become the benchmark for patience written into biology. Its pregnancy lasts approximately 22 months, a span so long that it often serves as the standard against which other prolonged gestations are measured. Entire seasons arrive and depart before a calf is born. Yet far beneath the ocean surface, another animal stretches that timeline far beyond what seems intuitively possible. The frilled shark is estimated to remain pregnant for approximately 42 months, 3.5 years. No confirmed vertebrate is known to exceed it. The number appears almost disconnected from ordinary experience. A child can learn to walk, speak in simple sentences, and begin forming lasting memories within that same span of time. Entire research projects begin and conclude. Ships cross oceans countless times. Meanwhile, within the body of a female frilled shark, development may still be unfolding at a pace that seems detached from the rhythms familiar to life nearer the surface. This extraordinary estimate emerged from the work of researchers in Japan who examined pregnant females recovered through deep water fishing operations. Because no scientist has ever observed a complete frilled shark pregnancy from conception through birth, direct measurement has never been possible. Instead, investigators relied upon embryo size and developmental stage. By comparing embryos from different females and calculating growth rates, researchers estimated that development proceeds at roughly 1.4 centimeters per month. Working backward from the size of near-term embryos produced a gestational timeline approaching 42 months. The figure remains an estimate rather than a directly observed fact, yet it has been repeatedly cited throughout the scientific literature because no better explanation currently fits the available evidence. The deeper significance lies not in the number itself, but in what creates it. Everything within the deep ocean moves according to a different metabolic rhythm. Cold water slows biological processes across nearly every level of organization. Cells divide more gradually. Proteins are assembled more slowly. Growth proceeds with reduced urgency. Chemical reactions continue, but at a pace shaped by an environment where conserving energy matters more than accelerating development. Embryos are not exempt from these constraints. The developing frilled shark grows within a world already adapted to slowness. Its tissues form according to a timetable established not by seasonal abundance or rapid productivity, but by cold stability. The environment surrounding the mother influences every stage of development, stretching the process across years rather than months. The reproductive strategy itself adds another layer to the story. Frilled sharks practice lithotrophic viviparity. The embryos develop inside the mother and are born alive, yet they do not receive nourishment through a placenta. Instead, each embryo carries its own yolk reserve, a self-contained source of nutrition that sustains growth throughout development. The mother provides protection and an appropriate internal environment, but the embryo remains largely responsible for its own nourishment. Growth depends on resources packaged at the beginning of development and consumed gradually over an exceptionally long interval. Litter sizes typically range from 2 to 15 pups, with approximately 6 often cited as an average. By the standards of many fishes, this is modest. For a species investing years into each reproductive cycle, every offspring represents a substantial commitment of time and biological resources. Such a strategy carries consequences. A population built upon long pregnancies, relatively small litters, and delayed maturity cannot respond rapidly to losses. If adult numbers decline, recovery unfolds slowly because replacement generations arrive at a measured pace. The effects of sustained pressure may remain hidden for years before becoming visible in population trends. Time itself becomes a limiting factor. The frilled shark cannot accelerate its reproductive schedule simply because conditions demand it. Its biology was shaped within an environment where urgency offered little advantage. Development proceeds according to rhythms established over immense spans of evolutionary history. A female conceiving a litter at the beginning of this story would still be carrying those embryos long after these words had faded into memory. Before that litter entered the world, this documentary could be viewed hundreds of times by entirely different audiences, each separated by months or years, while the same pregnancy continued its gradual journey toward completion in the darkness below. Birth is often imagined as a beginning protected by care. In many animals, the first days of life unfold beneath the attention of parents, within nests, dens, herds, or family groups. Knowledge is acquired through observation. Mistakes are buffered by protection. Survival begins with assistance already waiting. The frilled shark enters the world under different terms. A newborn typically measures between 40 centimeters and 60 centimeters in length. Even at the smaller end of that range, it is already larger than many fully grown bony fish inhabiting the same depths. Its body is not a fragile larval form waiting to transform into something capable. It arrives with the general shape, proportions, and equipment that will define the rest of its life. The transition from embryo to independent predator occurs abruptly. Once birth takes place, the mother's role is finished. There is no feeding afterward, no guarding of offspring, no transfer of territory, no escort through dangerous waters, no demonstration of hunting behavior. The immense investment represented by years of gestation concludes in a single moment, and the young shark immediately becomes responsible for itself. The yolk sac that sustained development has already been absorbed. Nothing remains to provide an extended reserve after birth. There is no protected interval during which survival skills can be acquired gradually. The newborn must begin locating prey and navigating its environment using the biological tools already present within its body. Fortunately, those tools are extensive. The sensory systems discussed earlier are not incomplete juvenile versions awaiting maturation. The lateral line is present. The electroreceptive organs, known as the ampullae of Lorenzini, are present. The olfactory system is present. The architecture needed to interpret pressure changes, electrical signals, and chemical traces already exists from the first moments of independent life. The dentition is equally remarkable. The young shark possesses the same fundamental retention system found in adults. Rows of inward curving teeth line the jaws, prepared to secure prey that ventures too close. The scale differs from that of a mature individual, but the design is already established. Nature does not provide a simplified practice model. It provides the complete blueprint immediately. What the newborn lacks is experience. It has never observed an adult hunting. It has never followed a parent through the water column. It possesses no memory of successful prey capture and no familiarity with the particular region of ocean it now inhabits. Every behavioral tendency available to it originates from inherited biological programming rather than learned observation. Even birth itself remains largely hidden from science. No researcher has directly witnessed a frilled shark giving birth in its natural environment. Information about newborn size and developmental condition comes primarily from near-term embryos recovered from pregnant females caught incidentally in deep water fishing operations. The earliest moments of independent existence remain unseen. This absence of observation extends beyond birth. Juvenile frilled sharks appear surprisingly rarely within the scientific record. Adult specimens are uncommon enough, yet juveniles are rarer still. Researchers have proposed several possible explanations. Young individuals may occupy specific microhabitats, seldom intersected by fishing gear. They may inhabit somewhat different depth ranges than adults. Mortality during the earliest life stages may also remove many individuals before they reach larger sizes. The available evidence does not yet permit a definitive answer. Whatever the explanation, the environment confronting a newborn is far from empty. Other predators share the same broad water column. Goblin sharks patrol portions of the deep ocean. Six-gill sharks move through overlapping regions. Large cephalopods hunt within the darkness. A young frilled shark measuring 50 centimeters does not occupy the highest position within this ecosystem. It enters a world containing both prey and predators from the beginning. And so a newly born animal drifts within water, measuring 4 degrees Celsius or 39 degrees Fahrenheit, under pressures approaching 50 atmospheres. Around it stretches a landscape without guidance, instruction, or companionship. Within it resides a design refined across immense spans of evolutionary history. Every sensory system, every tooth, every instinct has already been assembled. For that young shark, 80 million years of inherited engineering is not merely an advantage. It is the only map available. Some animals become rare because they inhabit only a single valley, a single island, or a narrow strip of coastline. Their scarcity makes intuitive sense. Geography confines them. Their world is small, and so their presence is small as well. The frilled shark presents the opposite situation. Its range stretches across much of the planet. Confirmed records from the Atlantic Ocean include specimens recovered off Portugal, Norway, Madeira, portions of West Africa, and the eastern coastline of the United States. Across the Pacific Ocean, the species has been documented near Japan, Australia, New Zealand, and Chile. Additional records emerge from multiple regions of the Indian Ocean, expanding the picture even further. Viewed on a global map, the pattern is striking. This is not a regional specialist restricted to one ocean basin or a single continental margin. It is a cosmopolitan deep-sea predator, a species whose confirmed presence spans every major ocean basin on Earth. Few sharks possess a distribution so extensive, yet the map tells only part of the story. The frilled shark does not occupy these oceans evenly. Its distribution follows particular underwater landscapes, features hidden beneath the surface, but enormously important to life in the deep sea. Continental slopes form one of the most significant of these environments. Along these regions, the seafloor drops steeply from the shallower continental shelf into deeper water, creating corridors where oceanographic conditions often concentrate prey. Submarine canyons appear important as well. These deep cuts into the seabed channel currents and transport organic material from shallower regions into greater depths. Energy and nutrients become focused rather than dispersed, attracting the organisms that form the foundation of local food webs. Seamounts provide another recurring feature. Rising from the abyssal plain like submerged mountains, they interrupt water flow and generate localized areas of enhanced productivity. Currents forced upward around these structures can create conditions favorable to prey species, indirectly benefiting predators occupying the same environment. The global picture became even more intriguing when researchers formally described a second species in 2009. Known as Chlamydosalacus africana, this relative is currently associated primarily with waters near South Africa and Angola. Its recognition demonstrated that the lineage contains more diversity than previously assumed. For decades, the frilled shark had been viewed largely through the lens of a single species. The discovery suggested that scientists were still assembling even the most basic taxonomic framework surrounding these animals. The genetic story remains similarly incomplete. Researchers cannot yet determine with confidence whether frilled sharks, distributed across distant oceans, belong to a single interconnected population, or whether multiple regional populations exist with limited exchange between them. Resolving that question requires extensive genetic sampling from locations scattered across the globe. Such work has only begun. This leads to one of the most remarkable paradoxes in deep-sea biology. The frilled shark occupies an enormous geographic range. It inhabits depths routinely reached by modern research equipment. It has been known to science for more than 140 years. By many measures, it should be a familiar animal. Instead, it remains one of the least observed sharks on Earth. The total number of confirmed live observations in natural habitat remains surprisingly small. Individual encounters are notable events. Researchers often discuss them in detail because each observation contributes meaningfully to a sparse body of direct evidence. Several explanations have been proposed. The species may exist at genuinely low population densities across its vast range. It may favor highly specific microhabitats that surveys do not consistently target. Certain behaviors may cause it to avoid or ignore equipment in ways that reduce detection rates. It may also spend substantial time at depths that fall between, above, or below the zones most commonly sampled by research programs. At present, none of these explanations has been confirmed. The mystery persists despite decades of technological progress and scientific effort. Vast distribution and persistent elusiveness coexist within the same animal. The contradiction remains unresolved. What endures is a peculiar reality. The frilled shark inhabits nearly every major ocean we have explored, follows underwater landscapes we understand reasonably well, and occupies regions visited by modern instruments. Yet even now, when scientists search the places where it should be, they often find only absence and the lingering possibility that somewhere nearby, unseen within the depths, it is already there. History is often measured in discoveries. A species is found, described, catalogued, photographed, observed, and eventually understood. Knowledge accumulates in layers, each generation adding detail to the work of the last. For many animals, that process unfolds steadily. For the frilled shark, it unfolded in a way that seems almost impossible. The scientific story begins in 1884. That year, Samuel Garman formally described Chlamydococcus sanguineus from specimens collected near Japan. The animal entered the scientific record with a name, a place within biological classification, and a growing collection of anatomical notes. From that moment onward, science officially knew the frilled shark existed. What science did not know was how it lived. For more than a century after the original description, nearly every specimen arrived in the same condition. It emerged from the depths aboard fishing gear, damaged by rapid ascent and exposed to environmental changes its body had never evolved to endure. Pressure dropped dramatically. Temperature shifted. Physiological systems adapted to deep water began to fail. Researchers could measure organs, count teeth, examine cartilage, and compare skeletons. They could learn what the animal was. They could not easily learn what the animal did. Generation after generation of marine biologists inherited essentially the same limitation. The subject of study was usually dead, or in the process of dying, before meaningful behavioral observation became possible. Then, after roughly 120 years of anatomical knowledge, something remarkable happened. In 2004, a Japanese deep-sea research submersible recorded the first confirmed footage of a living frilled shark in its natural environment. For the first time since the species entered science, the animal was observed where it actually belonged. The footage was brief. The shark moved slowly through the water, offering researchers a glimpse of natural locomotion. Important details were confirmed. Certain assumptions appeared reasonable. Yet the recording revealed only a fraction of what scientists hoped to understand. Feeding behavior remained unseen. Interactions with prey remained unseen. Nearly every major question surrounding daily life remained unanswered. Even so, the moment marked a turning point. 3 years later, another encounter captured public attention. In 2007, a live frilled shark reached the surface near Awashima Marine Park in Japan. The animal was filmed in captivity, producing footage that circulated widely and introduced many people to a creature that seemed almost prehistoric. Its elongated body, unusual gill structure, and unsettling appearance quickly became recognizable. The shark survived only a short time. Like many deep-sea species brought rapidly to the surface, it struggled under conditions fundamentally different from those in which it evolved. Within hours, it was dead. Subsequent years produced additional encounters, though only in small numbers. A handful of bycatch events brought living specimens briefly into view. Several submersible sightings added fragments to the growing record. Footage recorded in Japan during 2022 spread rapidly across the internet, once again reminding the public that this animal was not a fossil or legend, but a living species still inhabiting modern oceans. Yet the larger pattern remained unchanged. Researchers deployed baited camera systems within known frilled shark habitat, equipment operated for long periods in places where the species should theoretically appear. The results were often disappointing. Hours of recording frequently produced nothing at all. Occasionally, a fleeting pass through the frame rewarded the effort. The accumulated archive remains astonishingly small. When considering only footage showing living frilled sharks behaving in ways that might approximate natural behavior, the total is measured in minutes rather than hours. Even within those precious minutes, many individuals appear stressed, injured, disoriented, or affected by circumstances created through capture or human presence. The contrast becomes striking when viewed against the broader achievements of marine science. During the same span of approximately 140 years, researchers have attached satellite transmitters to individual great white sharks and followed their movements across entire ocean basins. Whale shark gatherings have been documented in extraordinary detail. Bottlenose dolphin societies have been studied closely enough to reveal complex social relationships spanning generations. The frilled shark remains different. 140 years have passed since its formal description. Entire scientific disciplines have transformed during that interval. Technologies once unimaginable now explore the ocean routinely. And still, for one of the oldest surviving shark lineages on Earth, humanity possesses only a few minutes of meaningful footage and a vast expanse of unanswered behavior hidden beyond the reach of observation. There is a difference between knowing what an animal is and knowing how it lives. For the frilled shark, that difference has become one of the most unusual scientific problems in modern marine biology. Across more than a century of research, scientists have assembled an increasingly detailed picture of the animal's anatomy. They know the shape of its skull, the arrangement of its organs, the structure of its teeth, the proportions of its body, and the contents of many stomachs recovered from captured specimens. Entire scientific papers have been built from these observations, each contributing another piece to the puzzle. Yet nearly all of that information originates from specimens that arrived at examination already dead, actively dying, or experiencing severe physiological distress. The pattern has repeated itself for generations. A frilled shark is brought upward from depths where cold temperatures and immense pressure form the baseline conditions of existence. Within a relatively short period, it is transported into an environment its body was never designed to experience. Pressure collapses towards surface levels. Temperature rises. Biological systems adapted to deep water begin responding to circumstances far outside their normal operating range. From such specimens, anatomy can be studied with remarkable precision. Structure leaves traces that persist after death. Researchers can measure fin dimensions. They can map muscle attachments. They can examine reproductive organs, count vertebrae, analyze stomach contents, and compare individuals from different regions of the world. These observations provide valuable information, and much of what is known about the species depends upon them. Function is another matter entirely. A preserved jaw can reveal how far it might open. It cannot reveal exactly how that motion unfolds during a real strike. Sensory organs can be mapped and measured. Their moment-to-moment operation while tracking prey through deep water remains hidden. The shape of the body may suggest a particular hunting posture, but suggestion is not observation. The proposed coiling strike remains a hypothesis because no one has witnessed it occurring in nature. The difficulty extends beyond visible behavior. Blood chemistry changes rapidly under stress. Hormone levels shift, neural activity patterns alter, immune responses become distorted. Metabolic measurements collected from an animal experiencing physiological crisis cannot be assumed to represent normal conditions. Once those changes begin, no later analysis can fully reconstruct what existed before them. The few living frilled sharks observed by humans have presented a similar challenge. Most appeared only after capture or during circumstances likely associated with severe distress. An animal struggling to survive unfamiliar conditions is not necessarily displaying the behaviors that define its ordinary life. The distinction is important. Observation alone is not enough. Context determines what the observation means. Scientific papers addressing the species often acknowledge this limitation directly. Their language carries a particular rhythm. Terms such as likely, possibly, suggests, and hypothesized appear frequently. These are not signs of weakness. They are signs of honesty. Researchers are marking the boundary between evidence and inference. Beyond that boundary lies an extensive territory of uncertainty. The coiling strike remains hypothesized. The effective range of electroreception is estimated through comparison with related species. Lifespan remains unknown. Population size has not been determined. Mating behavior has never been documented. Social interactions, if they exist at all, remain entirely unrecorded. When all of the certainty is gathered together, the resulting list is surprisingly short. The frilled shark inhabits cold, deep water. It consumes squid and other prey. It reproduces slowly, carrying developing young for an extraordinarily long period. It is encountered only rarely, despite its broad distribution. Almost everything else exists somewhere along a spectrum between informed inference and unanswered question. This does not represent a failure of science. It represents science functioning exactly as it should. Faced with an animal living beyond routine observation, researchers have resisted the temptation to claim certainty where certainty does not exist. After more than 140 years of study, the most accurate portrait of the frilled shark remains one drawn with careful lines around vast spaces that have yet to be seen. Sometimes a story travels around the world before anyone has time to check whether it is true. The deep ocean rarely enters public conversation, but when it does, it often arrives carrying a sense of mystery large enough to cross every language barrier. That happened in 2022. Footage of a frilled shark captured in waters near Japan spread rapidly across global media platforms. Within days, millions of people had watched the animal move across their screens. News articles appeared across continents. Social media feeds filled with reposted clips. For many viewers, it was the first time they had ever seen a frilled shark at all. The reaction was immediate. The animal's elongated body, ancient appearance, and unfamiliar anatomy seemed almost designed for modern headlines. Across hundreds of publications and countless online posts, a particular phrase appeared again and again. The frilled shark was described as a prehistoric, 80-million-year-old shark caught alive. The wording was memorable. It was also wrong. Not long after the footage spread, science communicators and researchers began issuing clarifications. Publications including British Broadcasting Corporation, Science Focus, explained the misunderstanding directly. The individual animal in the footage was not 80 million years old. No frilled shark lives for anything close to that length of time. Like other large marine animals, individual lifespans are measured in years and decades, not geological ages. The number belonged to the lineage. The distinction may seem small at first glance, yet it changes the entire meaning of the story. An 80-million-year-old individual would be something almost mythical, a creature somehow preserved unchanged across vast stretches of planetary history. The real frilled shark is something very different. It is an ordinary living animal, born, growing, reproducing, and dying within the rhythms of its own lifetime. What makes it extraordinary is not immortality. What makes it extraordinary is continuity. Its ancestors occupied deep ocean habitats tens of millions of years ago. Generation followed generation, each shaped by natural selection, each responding to the demands of a remarkably stable environment. Across immense spans of time, the basic design proved effective enough that dramatic change was never strongly favored. Evolution did not stop. Evolution simply continued refining a form already well suited to its world. In a strange way, the corrected version of the story is more remarkable than the original myth. An animal frozen in time belongs to fantasy. An animal that remained broadly recognizable because its environment remained consistently suitable belongs to biology. One idea depends on impossibility. The other depends on adaptation operating successfully across millions upon millions of years. The viral moment revealed something else as well. The public fascination was genuine. Millions of viewers stopped scrolling long enough to look at an obscure deep-sea shark that most people had never heard of before. The response demonstrated a widespread curiosity about life beyond ordinary human experience. For decades, the frilled shark occupied a narrow corner of scientific literature and specialist discussion. Suddenly, enormous audiences wanted to know more. That curiosity carries value. Deep-sea biology often struggles for attention because the subjects remain distant from everyday life. Yet the reaction to the frilled shark suggested that the barrier may not be lack of interest. It may simply be lack of exposure. When people are given a glimpse into this hidden world, many discover that they want to keep looking. The popular framing focused on age, because age is easy to understand. Ancient things naturally attract attention, yet age alone is not the central story. The deeper truth is that the frilled shark found a place where its particular design continued to work. While continents shifted, climates changed, and entire groups of organisms appeared and vanished, the deep environment it occupied remained sufficiently stable for the lineage to persist. The shark did not survive because it was old. It survived because it remained suited to the world around it. Far below the surface, that world still exists. The same dark slopes, the same drifting prey, the same cold water continue shaping the lives of animals hidden from routine observation. The frilled shark remains there as well, not as a relic preserved from another age, but as a living participant in the present, carrying a history that stretches backward across 80 million years of uninterrupted survival. Words can be remarkably durable. Once a phrase enters public imagination, it often survives long after scientists have begun arguing over whether it still describes reality accurately. Few examples illustrate this better than the label most frequently attached to the frilled shark: living fossil. The expression traces back to Charles Darwin, who used it to describe organisms that appeared strikingly similar to ancient forms known from the fossil record. Over time, the phrase escaped scientific discussion and entered popular culture. It carried an appealing image: a creature somehow preserved from another age, walking or swimming through the modern world unchanged while everything around it transformed. For decades, the frilled shark has stood among the most commonly cited examples. At first glance, the description seems reasonable. Its elongated body, unusual jaws, and ancient lineage create the impression of an animal transported directly from a distant chapter of Earth's history. Fossil relatives bear a resemblance to forms living today. The overall body plan appears remarkably conservative when measured across immense spans of geological time. Yet beneath the surface of the phrase lies a problem. The term suggests evolutionary stillness. It implies that change has somehow stopped. Biologically, that cannot happen. Every sexually reproducing organism accumulates genetic variation generation after generation. Mutations occur, genes are reshuffled. Natural selection continues operating whether visible changes emerge or not. Life cannot step outside evolution. The process remains active as long as reproduction continues. The frilled shark is no exception. Research using molecular phylogenetic methods has provided direct evidence that genetic change continues within this lineage. A study published during 2020 examined patterns within the genome and found signs of ongoing molecular evolution. Mutations have accumulated through time at rates broadly consistent with those observed in other shark lineages. The genome is moving forward. What appears unusual is that much of this movement is not expressed through dramatic changes in external anatomy. This distinction is important because morphology and genetics operate at different levels of biological organization. A genome may continue changing while the overall structure of the body remains broadly similar. These realities do not contradict one another. They simply describe different aspects of the same organism. The frilled shark can therefore be evolving continuously while still looking familiar to a paleontologist comparing it with ancient relatives. The reason lies not in resistance to evolution, but in the way evolution works. Natural selection does not generate novelty for its own sake. It does not pursue change as a goal. Instead, it favors characteristics that improve survival and reproduction under existing conditions. When environmental pressures shift, organisms often change in response. When conditions remain relatively stable and an existing design continues performing effectively, large structural alterations may provide little advantage. In such circumstances, major anatomical change accumulates slowly. The deep ocean has played an important role in this story. Surface environments have experienced repeated upheavals across geological history. Ice ages advanced and retreated. Sea levels rose and fell. Atmospheric conditions changed. Entire ecosystems reorganized themselves. Extinction events reshaped the trajectory of life more than once. Far below those surface dramas, the physical conditions experienced by many deep-sea organisms remained comparatively consistent. Temperatures stayed low, darkness persisted, pressure remained immense. The fundamental challenges confronting deep-water predators changed far less dramatically than those affecting many shallow water species. Within such a setting, a successful body plan may remain successful for a very long time. The frilled shark, therefore, occupies an unusual position in the evolutionary imagination. It is neither frozen nor static. Its genes continue moving through time, generation after generation, responding to mutation, inheritance, and selection, exactly as evolutionary theory predicts. What remains comparatively stable is the outward solution. Perhaps the most useful image is not that of a fossil preserved unchanged, but of a runner continuing steadily along a path that requires few adjustments. Movement never stops. Progress never ceases. The stride simply remains effective. Calling the frilled shark a living fossil captures part of the story, but only part. the deeper reality is more interesting. This is not an organism abandoned by evolution. It is an organism for which evolution has spent millions upon millions of years, refining a design that continues to meet the demands of its world with remarkable precision. Imagine tracing a family tree backward, so far that familiar branches begin to disappear. Species fall away, genera vanish, entire groups shrink into a handful of surviving lines. Eventually, the crowded complexity of the modern world gives way to something older, simpler, and far more revealing. The frilled shark occupies one of those rare positions near the deep foundations of shark history. It belongs to a group known as hexanchiforms, an order that contains the frilled sharks and the cow sharks. Among living sharks, this lineage sits at or near the base of the evolutionary tree, meaning it's separated from other shark groups closer to the origin of the entire shark lineage than any other surviving order. In evolutionary terms, it represents one of the earliest branches still alive today. The group is remarkably small. Only 6 living species are currently recognized within the order. 2 are frilled sharks, 4 are cow sharks. No other shark order contains so few surviving representatives while also occupying such an ancient position within shark evolution. Yet despite sharing common ancestry, these animals do not look particularly similar at first glance. The cow sharks retain certain ancestral features, including the unusual arrangement of 6 or 7 gill slits. Beyond that, however, they appear much closer to what modern observers recognize as a shark. Their bodies are broader, their proportions are more conventional. They are generally stronger swimmers, often occupying shallower environments than their elongated relatives. Examples include the blunt-nose 6-gill shark and the broad-nose 7-gill shark, both animals that retain traces of ancient anatomy while still fitting comfortably within modern expectations of shark form. The frilled sharks followed a different path. Although frilled sharks and cow sharks share a common ancestor, their separation stretches back tens of millions of years. That span of time is long enough for substantial differences to accumulate. Each lineage adapted to its own ecological circumstances, preserving some ancestral characteristics while developing distinctive traits of its own. The result is a branch within a branch, an ancient division nested inside an even older one. For scientists, this makes hexanchiforms extraordinarily valuable. Studying these animals provides more than knowledge about a handful of rare species. It offers clues about what the earliest sharks may have been like before later innovations reshaped the body plan. Features that appear unusual today may once have been common among ancestral sharks. Anatomical arrangements that now seem specialized may actually preserve older conditions from which newer forms evolved. In that sense, these animals function almost like living reference points. They cannot recreate the distant past, but they can illuminate aspects of it. The scale of evolutionary separation involved is difficult to appreciate without comparison. The frilled shark and the great white shark are both unquestionably sharks. They share a common ancestry and belong to the same broader group of cartilaginous fishes. Yet the evolutionary distance between them is immense, representing hundreds of millions of years of independent history. A useful comparison comes from mammals. A human being and a mouse are both mammals, sharing many fundamental characteristics despite obvious differences. The relationship between a frilled shark and a great white shark involves a similarly profound level of separation. Familiar labels can sometimes conceal extraordinary evolutionary distances. This perspective changes the meaning of every anatomical feature discussed so far. The forward-facing mouth is not merely unusual. The 6 gill slits are not merely unusual. The elongated body, the rearward dorsal fin, and the distinctive proportions are not merely unusual. Each characteristic carries information from deep evolutionary history, preserving clues about stages in shark development that most surviving lineages no longer display. Seen through this lens, examining a frilled shark becomes something larger than studying a single species. It becomes an opportunity to look toward the foundations of shark evolution itself, toward forms that existed before the familiar silhouettes of modern sharks emerged. Features that appear strange to contemporary eyes may once have represented the ordinary condition from which countless later variations evolved. The frilled shark is therefore not simply an outlier among sharks. It is a reminder that every successful modern design began as something else, and that hidden within the oceans, there still survives a lineage carrying traces of what sharks were becoming, before most of them became something entirely different. Long before human beings appeared, before mammals became dominant, before flowering plants spread across continents, Earth repeatedly passed through intervals when life itself seemed uncertain. Entire ecosystems vanished. Vast branches of the evolutionary tree ended. Species that had flourished for millions of years disappeared forever, leaving only fragments of their existence preserved in stone. The frilled shark lineage has lived through all of it. Its history stretches across a period that includes every major mass extinction event occurring after the lineage emerged. While countless organisms appeared and vanished around it, this branch of shark evolution continued forward, generation after generation, through some of the most disruptive episodes in planetary history. Among those events, one stands above the rest. Approximately 66 million years ago, an asteroid struck Earth, triggering what scientists call the Cretaceous-Paleogene extinction event. The consequences extended across the globe. Non-avian dinosaurs disappeared. Entire marine groups vanished. Roughly 75% of all species were eliminated. The catastrophe began at the surface, but quickly propagated through connected systems. Dust, soot, and debris entered the atmosphere. Sunlight reaching Earth declined dramatically. Photosynthesis weakened. Food webs built upon photosynthetic productivity began collapsing from their foundations upward. Oceans felt the effects as profoundly as continents. Organisms dependent upon surface productivity found themselves caught within a chain reaction extending across entire ecosystems. Near the surface, the consequences were severe. In the deep ocean, the story unfolded differently. Depth could not prevent disruption entirely. The deep sea depends ultimately upon energy generated far above it. Organic material, drifting downward from productive waters, sustains much of the life below. When surface ecosystems faltered, the supply of food reaching deep habitats declined as well. Yet the deep environment possessed a degree of insulation. Distance separated it from immediate surface chaos. Immense pressure remained unchanged. Temperatures shifted far more slowly than conditions near the surface. Thermal inertia buffered the environment against sudden fluctuations. The catastrophe arrived, but it arrived filtered through kilometers of water, which is about 621 miles, and through ecological pathways that softened some of the most abrupt impacts. Evidence suggests the frilled shark lineage crossed this boundary successfully. Fossil teeth and other remains attributed to frilled shark relatives appear in geological layers, both below and above the boundary, marking the extinction event. The lineage existed before the catastrophe, endured through it, and continued afterward into a world profoundly altered from the one that came before. This was not the only crisis. An even older extinction event occurred approximately 201 million years ago. Known as the Triassic-Jurassic extinction, it eliminated roughly 76% of species. Although the oldest confirmed frilled shark fossils appear later, evidence suggests the broader Hexanchiformes lineage likely originated before or near this interval. The ancestral branch leading toward modern frilled sharks appears to have survived that upheaval as well. Across geological history, a pattern begins to emerge. Many extinction events concentrated their most destructive effects within surface environments. Continents, coastlines, shallow seas, and sunlit ecosystems often absorbed the greatest impacts. The deep ocean certainly experienced disruption, but frequently at reduced intensity. Ecological consequences still arrived, yet they arrived altered by distance and filtered through layers of environmental stability. That distinction matters. It is tempting to describe extinction survivors as tougher, stronger, or inherently superior to species that vanished. The fossil record rarely supports such simple narratives. Survival often depends as much upon circumstance as capability. An organism may persist not because it possesses extraordinary resilience, but because it inhabits a place where the worst consequences arrive diminished. The frilled shark lineage appears to fit that pattern remarkably well. Its survival record is not necessarily evidence of exceptional adaptability. Many highly adaptable species disappeared during mass extinctions, nor is it evidence of unusual strength. Geological disasters do not reward strength in any straightforward sense. Instead, the lineage benefited from where it lived. While catastrophe repeatedly swept across the surface world, the deep ocean absorbed those shocks differently. The disturbances still came, but they arrived weakened by depth, moderated by physical stability, and distributed across longer spans of time. In that sense, the frilled shark's survival through extinction after extinction is less a story about endurance than location. Its greatest evolutionary advantage may not have been what it was, but where it happened to be when the world changed. Some environments shape the creatures that inhabit them. Others seem to disappear into the background, providing a stage upon which evolution unfolds. The deep sea belongs to neither category. It is not merely a setting, it is an active force, a persistent architect, a system whose physical rules reach into every aspect of the lives that develop within it. The frilled shark is one of its clearest creations. It is tempting to think of the deep ocean as the place where this animal happened to survive. The relationship runs deeper than that. The environment did not simply shelter the frilled shark. It produced the conditions that made the frilled shark possible and sustained those conditions across spans of time almost impossible to imagine. Much of this begins with stability. At a given depth, pressure remains effectively constant. Darkness remains constant. Seasonal cycles that dominate surface ecosystems never arrive. Across geological intervals, temperatures within stable deep water masses may vary by less than 2 degrees Celsius or 36 degrees Fahrenheit. Conditions shift, but they often do so gradually and within narrow limits compared with the dramatic fluctuations experienced nearer the surface. For organisms living there, stability becomes an evolutionary force of its own. A lineage well adapted to such an environment has little incentive to leave it. Moving into a fundamentally different habitat would require extensive physiological adjustments. Sensory systems tuned to darkness might become less effective. Metabolic strategies optimized for scarce food could become liabilities elsewhere. Anatomical structures successful under one set of conditions might fail under another. Evolution always involves trade-offs. If an environment already supports survival and reproduction, abandoning it may cost more than remaining within it. Over long spans of time, this tendency encourages what biologists sometimes describe as habitat conservatism. Successful lineages remain where they are successful. The frilled shark embodies that principle. Its biology is often described feature by feature: a slow metabolism, an enormous liver, prolonged reproduction, ambush predation, hundreds of inward curving teeth. Yet these are not independent inventions assembled by chance. They function together as parts of a single integrated solution. Cold water reduces metabolic demands. Scarce food favors energy efficiency. Extended intervals between meals reward large energy reserves. Darkness favors sensory systems capable of detecting movement, electricity, and chemical traces. Elusive prey rewards mechanisms that prevent escape once contact is made. Each trait complements the others. Remove the environment and the design ceases to make sense. Place the design within that environment and its coherence becomes apparent. The pattern extends beyond a single species. The coelacanth, a lobe-finned fish whose ancestry reaches back approximately 400 million years, occupies similarly stable, deep habitats. The nautilus retains a body plan recognizable across immense spans of geological time. The vampire squid inhabits oxygen-poor waters where environmental conditions remain comparatively consistent. These organisms differ enormously in ancestry and appearance, yet certain themes recur: cold water, low metabolic rates, slow reproduction, long-term anatomical stability. The similarities do not arise because these species are closely related. They arise because similar environments often favor similar strategies. This perspective becomes even more striking when considering the scale of the deep ocean itself. Human attention naturally gravitates toward continents, coastlines, forests, grasslands, and surface waters. Yet, the deep ocean covers roughly 65% of Earth's surface and contains most of the planet's habitable volume. By sheer size, it is the dominant biome of the modern world. Its rules are not the rules of the surface. Energy arrives differently. Time operates differently. Change often unfolds more gradually. Biological success is measured according to conditions unfamiliar to terrestrial experience. Viewed through that lens, the frilled shark ceases to appear unusual. It is not an accident. It is not an exception. It is not a mistake, preserved by chance. It is the predictable outcome of an environment that rewards patience over speed, efficiency over excess, and stability over constant reinvention. Given enough time, the deep sea tends to shape life according to those principles. The frilled shark stands as one expression of that process, a lineage molded by darkness, pressure, scarcity, and persistence. What appears extraordinary from the perspective of the surface becomes entirely logical when seen through the eyes of the world that created it. A conservation assessment often begins with a simple question: how many animals are there? For the frilled shark, that question drifts into darkness almost immediately. Globally, the species is currently classified as least concern by the International Union for Conservation of Nature. At first glance, the designation appears reassuring. Yet, beneath that label sits an important qualification. In parts of the Northeast Atlantic and Northwest Pacific, regions where deep-sea commercial fishing activity is especially concentrated, the species is considered near threatened. The difference between those categories reveals something important about the limits of what is actually known. The global classification does not rest upon evidence demonstrating a stable population. It rests largely upon the absence of evidence demonstrating a decline. Those are not the same thing. The assessment itself acknowledges that available data remain insufficient to determine long-term population trends with confidence. Scientists know the animal exists across vast areas of the world. Ocean. They know it continues to appear in fishing records. Beyond that, certainty becomes increasingly difficult to obtain. Most conservation tools depend on visibility. Bird populations can be counted from the air. Large mammals can be photographed, tracked, and surveyed across landscapes. Coastal marine species can often be monitored through tagging programs, visual censuses, or repeated observations. The frilled shark inhabits a realm where such methods become extraordinarily difficult. Aerial surveys are impossible. Mark recapture studies are impractical because encounters are so infrequent and habitats so inaccessible. Acoustic monitoring remains largely untested for this species. Population models require baseline estimates that do not yet exist. Even establishing whether a regional population is increasing or decreasing becomes a formidable challenge when the starting number is unknown. Meanwhile, the pressures acting upon the species are easier to identify than the populations experiencing them. The primary threat comes not from targeted fishing, but from bycatch. Deep-sea commercial trawlers searching for valuable species such as grenadiers, orange, ruffy, and deep-water groupers often operate within the same depth ranges occupied by frilled sharks. Nets intended for one animal occasionally return another. For a frilled shark, survival after capture is extraordinarily unlikely. An organism adapted to cold water and immense pressure cannot easily withstand a rapid journey to the surface. Barotrauma, temperature change, and severe physiological stress act together. By the time the animal reaches a vessel, the outcome is usually already determined. Catch and release, a conservation tool effective for many other species, offers little benefit here. The challenge extends beyond individual animals. Bottom trawling physically alters the landscapes through which these sharks move. Continental slopes and submarine canyons contain ridges, sediment structures, and complex terrain that influence deep-sea ecosystems. Repeated contact with heavy fishing gear can simplify those environments, reducing structural complexity and reshaping habitats that took long periods to develop. Protection efforts face additional complications. Deep-sea fisheries operate under a patchwork of regulations that vary among nations, management organizations, and protected areas. Some regions impose restrictions on vulnerable habitats, others do not. Policies often reflect economic priorities, regional circumstances, and differing levels of scientific information. Within that system, the frilled shark occupies an unusual position. It is not commercially valuable enough to become the focus of intensive fisheries management, yet it is not prominent enough to attract widespread conservation attention. Existing largely outside public awareness, it occupies a narrow space between regulation and neglect. All of this compresses into a difficult reality. Effective conservation normally begins with knowing how many animals exist, where they are found, and whether their numbers are changing. For the frilled shark, each of those foundations remains uncertain. The species lives in a habitat that is exceptionally difficult to monitor, encounters fishing activities directed toward entirely different targets, and reproduces at a pace measured over years rather than seasons. Any decline unfolding today may remain hidden for a very long time, carried through the deep ocean beyond observation, detectable only after the consequences have already traveled far ahead of the evidence. Sometimes the most revealing part of a scientific document is not what it claims to know, but what it openly admits it cannot know. The conservation assessment for the frilled shark carries that quality throughout its pages. Beneath the formal language and categorical rankings lies an unusual degree of candor. Rather than presenting a complete picture of the species, the assessment repeatedly circles the boundaries of uncertainty, marking the edges of knowledge with notable care. At the center of that uncertainty sits a classification that has long invited debate. Globally, the frilled shark is listed as least concern. On paper, the phrase sounds straightforward. It suggests a species that is not currently facing significant extinction risk. Yet within the framework used by the International Union for Conservation of Nature, that category traditionally assumes something important. It assumes sufficient information exists to evaluate the status of the population with reasonable confidence. Another category exists for species lacking that foundation. Data deficient is reserved for cases in which available information is too limited to determine risk. It does not mean a species is secure. It does not mean a species is threatened. It means the evidence is inadequate to support either conclusion. For many deep-sea biologists, the frilled shark occupies an uncomfortable position between those two definitions. Scientists know remarkably little about total population size. They do not possess reliable long-term population trends. They cannot confidently estimate recruitment rates, mortality rates, or regional abundance across most of the species' enormous range. In many respects, the informational gaps resemble the circumstances that often justify a data-deficient classification. The decision to retain least concern emerged from a different form of reasoning. In the absence of documented decline, assessors generally avoid assuming decline exists. The logic is understandable. Conservation categories are intended to reflect evidence. If no measurable reduction has been demonstrated, assigning a higher risk category becomes difficult to justify under existing standards. Yet that same logic carries an inherent vulnerability. A species living beyond routine observation may experience substantial change before sufficient evidence accumulates to document it. Declines that occur in inaccessible environments can remain hidden for long periods. By the time they become measurable, much of the process may already have unfolded. The contrast becomes clearer when viewed alongside other sharks. The school shark has been classified as critically endangered in parts of its range. The gulper shark is considered vulnerable in many assessments. Several deepwater dogfish species occupy categories ranging from near threatened to endangered. Those determinations emerged from fisheries records, population analyses, and bycatch datasets considerably more extensive than the information available for the frilled shark. The comparison does not necessarily imply that the frilled shark faces equivalent danger. It does, however, highlight a fundamental asymmetry. Some species are assessed using imperfect but substantial evidence. The frilled shark is often assessed using evidence that is sparse from the outset. Even the bycatch record may conceal complexities. A rare appearance in fishing gear can be interpreted in several ways. It may indicate genuinely low abundance. It may indicate that the species occupies habitats only occasionally intersected by commercial operations. Submarine canyons, steep continental slopes, and highly specific terrain features may concentrate animals in places that surveys rarely reach. Under such circumstances, rarity in the data can become difficult to distinguish from rarity in nature. This challenge has been acknowledged repeatedly by specialists studying deep-sea sharks and their relatives. The problem is not simply that more observations are needed. The problem is that many existing methods were designed for environments where repeated observation is possible. Applying those same tools to deep ocean species often reveals their limitations. Viewed from that perspective, the assessment becomes something more than a statement about the frilled shark. It becomes a reflection of measurement itself. The category attached to the species tells part of the story. The uncertainty surrounding that category tells another. Together, they reveal a reality that extends beyond a single shark, pointing toward a broader truth of deep ocean science. Sometimes the greatest obstacle is not the condition of the animal being measured. It is the difficulty of measuring it at all. There are animals whose lives become easier to understand once they enter human care. A forest bird can be observed in an aviary. A large shark can be studied within an oceanarium. Even species from difficult environments sometimes reveal their biology when transferred into carefully controlled facilities. The frilled shark has refused every such attempt. Across the entire history of documented encounters, no institution has succeeded in maintaining a living frilled shark for more than a brief span of time. The record is complete in its consistency. Animals brought to the surface die within hours, despite intervention, monitoring, and efforts to recreate aspects of their natural environment. The failure is not the result of neglect or inadequate preparation. It begins long before the animal reaches a tank. Far below the surface, the frilled shark exists within a world defined by immense pressure. At depths around 500 meters, which is about 311 miles, every part of its body functions under physical conditions fundamentally different from those found at sea level. Blood chemistry, tissue mechanics, cellular processes, and dissolved gases all exist within an equilibrium established by that pressure. When ascent begins, that equilibrium starts to unravel. Gases held in solution within body fluids behave differently as pressure decreases. They expand, separate, and move through tissues in ways that can cause extensive damage. Blood vessels may be affected. Organs may experience disruption. Internal injuries can develop throughout the body. The process resembles severe decompression sickness in human divers, although the physiological details differ because the frilled shark evolved under conditions far more extreme than any routinely experienced by people. The damage unfolds rapidly. Commercial trawling equipment is designed to retrieve nets efficiently, not to provide gradual decompression over extended periods. By the time a captured shark reaches the surface, multiple biological systems may already be failing. Available technology offers little opportunity to reverse the cascade once it has begun. Temperature introduces another burden. Deep water occupied by frilled sharks commonly remains near 4 degrees Celsius or 39 degrees Fahrenheit. Surface conditions are often substantially warmer. Even when handlers attempt to reduce thermal stress, the transition itself places intense demands on an animal whose enzymes, tissues, and metabolism are adapted to near freezing water. Physiological systems calibrated for one environment are suddenly forced to operate within another. The combined effects can be overwhelming. One of the most widely documented examples occurred at Awashima Marine Park in 2007. Staff responded with care and urgency. Water conditions were adjusted. Temperature was managed as effectively as possible. Every reasonable measure available at the facility was employed. None altered the outcome. The animal died within hours. That event was not an exception. It was another entry in a pattern that has repeated whenever living specimens have reached human hands. Researchers have achieved remarkable successes with some deep-sea organisms. Specialized, pressurized aquarium systems have allowed certain species to survive under conditions approximating their natural habitats. Advances in engineering continue to expand what is possible. Yet, even with those developments, frilled sharks have never been maintained successfully. This suggests that pressure and temperature, although critical, may not be the only factors involved. Additional physiological requirements may exist that remain poorly understood. That reality creates an unusual ethical tension. Every live frilled shark recovered from a net presents a difficult choice. Scientific examination can produce valuable information, yet obtaining that information comes at a cost that is effectively predetermined. The animal will not return to its habitat. The specimen, the measurements, and the observations are acquired through a process that ends with death. As a result, the future of frilled shark research points away from captivity rather than toward it. There will be no thriving colony in a research aquarium, no long-term behavioral studies conducted behind glass, no controlled experiments revealing daily routines through direct observation. The living biology of the species must be approached differently, through technologies capable of meeting the animal within its own world rather than bringing it into ours. In that sense, the frilled shark imposes its own conditions upon science. To understand it, researchers must learn how to observe without possession, how to study without removal, and how to gather knowledge while leaving the animal where it has always belonged. At this point in the story, after fossils and anatomy, after distribution maps and conservation assessments, after more than a century of papers, specimens, and investigation, it becomes possible to perform an unusual exercise. Rather than listing what science knows about the frilled shark, we can list what it does not know. The result is longer than many people expect. Population size remains unknown, not approximately known, not debated between competing estimates, not uncertain within a broad range, unknown. No accepted estimate exists for any major ocean basin. There is no global population figure. There is currently no proven methodology capable of producing one with confidence. The number of frilled sharks alive on Earth today remains absent from the scientific record. Lifespan remains unknown. Researchers suspect that individuals may live for decades, based partly on comparisons with other sharks possessing similarly slow life histories. Yet no age determination method has been successfully applied to frilled sharks on a meaningful scale. The true lifespan of the species has not been established. Hunting behavior remains almost entirely undocumented. The proposed coiling strike, discussed throughout scientific literature, remains exactly what it has always been, a hypothesis. No feeding event has ever been filmed in the natural environment. No direct observation has confirmed how prey is approached, captured, or subdued under ordinary conditions. Movement patterns remain unknown. No frilled shark has ever been successfully tagged and tracked through time. Scientists cannot say whether individuals occupy relatively small home ranges or undertake extensive journeys. Seasonal movement patterns remain unrecorded. Whether populations from distant regions exchange individuals across vast stretches of ocean is also unresolved. Social behavior remains unknown. The species is often described as solitary, but that description emerges largely from a lack of observations, rather than from observations demonstrating solitude. Researchers do not know whether individuals interact regularly, gather during reproduction, or display any recurring social patterns at all. Mating behavior remains entirely undocumented. No courtship has been observed. No mating event has been recorded. The mechanisms through which males and females locate one another across immense volumes of deep water remain uncertain. Even the environmental cues that initiate reproductive activity are unknown. The early lives of young, frilled sharks remain largely hidden. Scientists possess information about developmental stages before birth and scattered information from rare juvenile specimens. Beyond that, the record becomes remarkably thin, where juveniles spend their first years, how quickly they grow, what prey they target, and what predators threaten them remain questions with few reliable answers. Population structure remains unresolved. The species occurs across multiple ocean basins, yet researchers cannot determine whether those occurrences represent one broadly connected population or several regional populations separated by geography and depth. Genetic sampling has begun to explore the issue, but definitive conclusions remain out of reach. Even reproduction contains uncertainties beyond the famous estimate. The commonly cited gestation period of approximately 42 months represents an inference drawn from available evidence. It is the best estimate currently available. It is not a directly observed measurement. The exact duration has never been confirmed from beginning to end. Placed individually, each unknown appears manageable. Placed together, they create a different impression. 140 years have passed since the species entered the scientific literature. During that time, researchers have described its anatomy in extraordinary detail. They have mapped its distribution across the globe. They have compared specimens, examined tissues, analyzed stomach contents, and reconstructed evolutionary relationships stretching deep into geological history. Yet the most basic elements of its living existence remain hidden. How many there are, how long they live, how they hunt, how they move, how they reproduce, how they spend their earliest years. The list requires no embellishment. It needs no dramatic interpretation. It stands on its own as a remarkably honest inventory of uncertainty, accumulated across a century and a half of study. The weight of that inventory reveals something profound, not about a failure of investigation, but about the scale of the world that still lies beyond observation, even now. For most of the frilled shark's scientific history, knowledge advanced only when an animal died. A specimen appeared in a net, measurements were taken, tissues were examined. Another fragment of the puzzle was added to the record. The process generated valuable information, yet it also imposed a limit. Nearly every answer depended upon direct physical access to a creature that seemed determined to remain beyond reach. Now, for the first time, a different approach is emerging. Instead of bringing the animal to science, science is beginning to travel into the animal's world. One of the most promising tools is environmental deoxyribonucleic acid analysis. Every organism leaves traces of itself behind. Tiny fragments of skin, mucus, waste products, and cellular material drift through surrounding water. These fragments contain genetic information. By collecting water samples from specific depths and filtering them in specialized laboratories, researchers can search for those genetic signatures without ever seeing the animal that produced them. The implications are profound. Environmental deoxyribonucleic acid surveys have already detected frilled shark genetic material in locations where visual searches found nothing. The animal remained unseen, yet evidence of its presence lingered within the water itself. A kind of biological fingerprint persisted after the owner had vanished into darkness. As sampling techniques improve, researchers may finally gain a clearer picture of distribution patterns across entire regions. Rather than deploying nets across enormous areas, scientists can collect water samples at different depths, along continental slopes, near submarine canyons, and around underwater mountains. Repeated surveys could reveal where frilled sharks are most likely to occur and how those patterns change through time, all without disturbing a single individual. At the same time, advances in underwater robotics are changing what direct observation can accomplish. Early remotely operated vehicles often produced significant noise, bright illumination, and conspicuous movement. Modern systems are becoming more refined. They move more slowly. Their acoustic signatures are reduced. Their cameras can function under far lower light levels than previous generations. The probability that equipment itself alters animal behavior may gradually decrease as these technologies improve. Another approach waits patiently rather than actively searching. Baited remote underwater video systems consist of cameras placed near carefully selected bait and left unattended for extended periods. Instead of pursuing animals, they allow animals to approach on their own terms. These systems have already transformed studies of many deep-sea sharks. Increasingly sophisticated deployments are now being directed toward habitats associated with frilled sharks, with researchers refining bait strategies, deployment depths, and recording durations. Tracking technology is advancing as well. Engineers are developing acoustic tags capable of withstanding the immense pressures found within frilled shark habitat. The remaining obstacle is not the electronics themselves, but the challenge of attaching them without causing harmful stress. If that problem can be solved, movement patterns that have remained hidden for generations may finally become visible. Listening may prove just as important as watching. Across the world's oceans, extensive networks of hydrophones already record underwater soundscapes. Some were established for naval purposes. Others monitor geological activity or environmental conditions. Within those recordings lie countless biological sounds that remain unidentified. If researchers can determine whether frilled sharks produce distinctive acoustic signatures, passive monitoring may become possible across enormous areas without any direct interaction at all. What makes these developments remarkable is not merely their sophistication, it is the philosophy behind them. For more than 140 years, the central obstacle of frilled shark science has been the difficulty of observing a living animal without harming it. The newest generation of tools addresses that problem directly. They seek information through presence rather than capture, through patience rather than intrusion. The result may be a transformation in understanding. Questions that have persisted across generations could begin yielding answers. Distribution, movement, population structure, and behavior may become clearer through methods that leave the animal undisturbed within the environment that shaped it. The knowledge gap that has defined the frilled shark for so long is not necessarily permanent. Across laboratories, research vessels, and deep ocean observatories, the instruments capable of narrowing that gap are already taking shape, extending human observation farther into the deep than ever before, while asking less of the creature being observed. There is a tendency in human thought to place importance wherever visibility is greatest. The animals most frequently observed become the animals most discussed. The ecosystems nearest to cities become the ecosystems most studied. The species that appear in photographs, documentaries, museums, and textbooks gradually come to occupy the center of our understanding. Everything beyond that illuminated circle often feels secondary, as though the unseen portions of the living world exist mainly as background. The frilled shark resists that assumption with remarkable persistence. It is not a small organism hidden in an isolated pond. It is not a rare insect confined to a remote valley. It is a large predatory vertebrate distributed across multiple oceans, occupying the largest habitat on Earth, descended from one of the oldest surviving branches of shark evolution. By any ordinary measure, it should be considered significant. Yet, despite that significance, it remains largely unknown. This tension creates something deeper than a biological puzzle. It creates a challenge to the way knowledge itself is often imagined. Many people grow up with the impression that humanity possesses a reasonably complete inventory of large life forms. The remaining discoveries are expected to be minor refinements, perhaps a new species of insect, a previously overlooked fish, or a subtle revision to an existing classification. The broad outlines of life on Earth seem settled. The frilled shark suggests otherwise. For the entirety of recorded human civilization, this animal has been present beneath the oceans. It moved through deep water while the first cities emerged. It persisted through the rise and fall of kingdoms, through the creation of writing systems, through voyages of exploration, through industrialization, through every scientific revolution that transformed humanity's understanding of the universe. Throughout all of it, the frilled shark continued hunting, reproducing, and surviving beyond human awareness. Even today, after decades of technological advancement, direct knowledge of its daily existence remains astonishingly limited. That realization points toward a larger truth about the deep ocean itself. The deep sea constitutes the majority of Earth's habitable volume. It is not a marginal environment. It is not a biological afterthought. It is the planet's dominant living space. Yet scientific estimates for the number of species inhabiting that realm remain extraordinarily broad, ranging from roughly 500,000 to as many as 10 million. Such a range is not a minor statistical disagreement. It is an acknowledgement that vast portions of the biosphere remain beyond reliable measurement. The uncertainty spans an order of magnitude. In practical terms, it means science cannot yet say with confidence whether millions of species remain undiscovered or whether the total number is substantially smaller. The gap between those possibilities is itself enormous. Within that context, the frilled shark occupies a fascinating position. It is what researchers sometimes call a known unknown. Its existence is confirmed. Its anatomy has been described. Its evolutionary relationships are broadly understood. Scientists know enough to recognize it as real, distinctive, and important. Yet surrounding that limited knowledge is a much larger territory of unanswered questions. Beyond the frilled shark lie organisms that have never been collected, lineages that have never been recognized, and ecological relationships that have never entered scientific literature. Entire ways of living may persist in darkness without names, classifications, or histories. The frilled shark becomes significant, not merely because of what it is, but because of what it represents. A large predator has existed across the world's oceans for tens of millions of years. For almost all of that time, humanity did not know it existed. Even now, humanity understands only fragments of its story. This is not evidence of failure. It is perspective. The planet is larger, older, and more biologically complex than human observation alone might suggest. The frilled shark stands as one of the clearest reminders of that reality, a living marker placed at the boundary between what has been documented and what remains beyond the reach of current knowledge, waiting within a world that is still far less familiar than it often appears. Far below the familiar geography of the surface, where continents end and descending slopes fade into immense open water, the frilled shark occupies a neighborhood that is far older and more complex than it first appears. It is easy to imagine such depths as empty. The reality is something different. The frilled shark moves through a living community, one populated by lineages whose histories stretch backward across astonishing spans of time. Many of its neighbors carry the same signature of persistence, bodies that have remained recognizable across geological ages because the environments that shaped them have changed so little. Among the closest of these neighbors is the blunt-nosed six-gill shark. Like the frilled shark, it belongs to the ancient order known as hexanchiforms. Their shared ancestry reaches deep into shark evolutionary history, to a time before many modern shark forms had emerged. The blunt-nosed six-gill follows a different path in appearance, possessing a broader body and a more familiar shark silhouette, yet traces of their common heritage remain visible. Reaching lengths of approximately 4.8 meters, which is about 15.7 miles, it ranks among the largest predators inhabiting the mesopelagic realm. Across overlapping depth ranges, both species continue lineages that diverged tens of millions of years ago while retaining echoes of a much older biological world. Not far above portions of the frilled shark's range swims another remarkable survivor, the goblin shark. Its elongated snout and extraordinary jaws create an appearance so unusual that it often seems almost fictional. Yet the goblin shark represents another ancient branch of shark evolution. Its protrusible jaws can surge forward with remarkable speed during feeding, demonstrating that extreme jaw mechanics evolved more than once in deep ocean environments. Separate evolutionary pathways arrived at similarly specialized solutions, shaped by conditions that reward efficiency in encounters where opportunities may be rare. The community extends beyond sharks. Drifting through oxygen-poor waters is the vampire squid, an animal whose name often creates misleading impressions. It is neither a true squid nor an octopus in the conventional sense, but the representative of an ancient cephalopod lineage occupying its own distinctive branch of evolutionary history. Fossil evidence suggests that relatives of this lineage existed long before many modern cephalopod groups appeared. Across immense stretches of time, its fundamental design has endured. Elsewhere in these depths, giant squid travel through darkness, vast enough to conceal even creatures of enormous size. Fragments of giant squid have been identified within frilled shark stomach contents, linking the two species through the ancient exchange of predator and prey. Yet the relationship is not necessarily one-sided. Smaller frilled sharks may themselves face danger from encounters with large cephalopods. The boundaries between hunter and hunted shift constantly in deep water, often hidden from observation. Surrounding all of them is a phenomenon that transforms the character of the environment itself. An estimated 76% of deep-sea organisms produce light through biological processes. Countless flashes, pulses, glows, and signals move through the darkness. The result is not a world illuminated like daylight, but a landscape animated by living points of radiance generated from within the organisms themselves. Communication, camouflage, attraction, and defense are written in brief sparks of biological light. Moving through this illuminated darkness is another recurring event, the deep scattering layer. Twice each day, vast numbers of mesopelagic organisms migrate vertically through the water column. Billions of animals rise and descend in one of the largest daily movements on Earth. For predators inhabiting these depths, the migration creates a predictable pulse of opportunity passing through an environment that rarely offers certainty. Seen within this broader context, the frilled shark becomes something different from a solitary relic. It is one member of an ancient community, surrounded by organisms whose histories, adaptations, and persistence reveal the character of the deep sea itself. What appears unusual from the perspective of the surface is often entirely ordinary here. In this hidden neighborhood, evolutionary conservation is not an exception. It is one of the defining features of life, woven through a living world that humanity has only begun to observe. Imagine standing at the edge of a map and discovering that the blank space beyond it is larger than everything already drawn. That is, in many ways, the relationship humanity has with the deep ocean. Beneath a depth of 200 meters, which is about 124 miles, begins a realm so vast that ordinary intuition struggles to contain it. This immense region covers approximately 65% of Earth's surface. When measured not as area but as three-dimensional living space, it represents roughly 95% of the planet's habitable volume. By scale alone, it is Earth's dominant biome. All forests, grasslands, deserts, rivers, lakes, mountains, wetlands, coastlines, and shallow seas occupy a comparatively smaller share of the living world. The environment most familiar to human experience is not the planet's primary habitat. It is simply the portion most accessible to human senses. Yet this largest habitat remains the least inventoried. It is also among the least monitored and least protected. Applied to a terrestrial environment, such a statement would seem astonishing. Imagine if most of the Amazon rainforest remained unsurveyed, if population estimates for major predators were unavailable, if large regions had never been systematically explored. Such circumstances would likely be treated as urgent scientific priorities. In the deep ocean, they are commonplace. The reason is not neglect alone, it is difficulty. Every descent into deep water demands specialized equipment, research vessels, engineering support, and substantial funding. A remotely operated vehicle expedition reaching 1,000 meters, which is about 621 miles, may cost more than many terrestrial wildlife studies while covering only a tiny fraction of the area that land-based researchers can survey in the same period. The economics of exploration have imposed limits that technology is only now beginning to challenge. As a result, uncertainty remains embedded in even the most basic questions. Scientists estimate that the deep ocean may contain somewhere between 500,000 and 10 million species. The range is enormous. It spans an order of magnitude. Such breadth does not exist because researchers are careless with numbers. It exists because the unknown portion of the system is genuinely that large. Many organisms have never been collected. Others have been collected but not formally described. Still others may remain entirely beyond present observation. The physical landscape itself reflects the same vastness. Running across the ocean floor is the mid-ocean ridge system, the longest mountain chain on Earth. Stretching for tens of thousands of kilometers, which is about tens of thousands of miles, it winds around the globe beneath the sea, largely hidden from public imagination despite exceeding the scale of any continental mountain range. Elsewhere, Hadal trenches descend to depths approaching 11,000 meters, which is about 6,835 miles. Hydrothermal vent systems support entire ecosystems, fueled not by sunlight, but by chemical energy emerging from the planet's interior. Life flourishes there, according to rules that would seem extraordinary if encountered for the first time. Against this backdrop, the frilled shark occupies only a portion of the deep ocean. Its range lies primarily within the mesopelagic and upper bathypelagic zones. These are not even the deepest environments available. Vast territories extend far below them, descending into regions even less explored than the waters the frilled shark calls home. And yet, The frilled shark remains this mysterious. Its story reveals something larger than the biology of a single species. The unanswered questions surrounding its behavior, distribution, reproduction, and population structure are not anomalies. They are symptoms of a broader reality. The deep ocean contains immense living systems that continue functioning beyond routine human observation. Entire ecological relationships unfold without documentation. Countless organisms live and die without ever entering scientific records. The frilled shark becomes a symbol of that hidden majority. Not because it is uniquely mysterious, but because it is one of the rare inhabitants of the deep sea, large enough for people to recognize and still difficult enough to understand. Through it, a larger truth becomes visible. The planet is greater than the portion humanity has mapped, measured, and explained. Nowhere is that truth more apparent than in the deep ocean, where most of Earth's living space continues to exist beyond the boundaries of familiar knowledge. Most discussions of survival are built around change. The species that adapt fastest, innovate most effectively, expand into new environments, or outcompete rivals are often presented as evolutionary success stories. Survival becomes associated with transformation, as though persistence can only be achieved through constant reinvention. The frilled shark offers a different perspective. Across approximately 80 million years, its lineage has endured not because it continually became something new, but because it found circumstances in which what it already was remained sufficient. This distinction matters. Evolution is often imagined as a ladder ascending toward greater complexity, greater sophistication, or greater capability. In reality, evolution has no destination. It does not reward novelty for its own sake. It rewards whatever successfully produces surviving offspring within a particular environment. For the frilled shark, that environment happened to be one of the most stable on Earth. While continents shifted, climates oscillated, sea levels rose and fell, and ecosystems reorganized themselves repeatedly, the deeper ocean changed more gradually. Conditions varied, certainly, but not with the relentless intensity that characterized many surface habitats. The rules governing survival remained comparatively consistent across immense stretches of time. Within those conditions, the frilled shark's design continued to work. Its physiology remained effective. Its feeding strategy remained effective. Its reproductive approach remained effective. The integrated collection of traits that define the species kept solving the same ecological problems generation after generation. There was no requirement to become something else. Seen from this perspective, 80 million years is not merely a span of time, it is evidence. A design that persists across such a duration has been tested against countless environmental fluctuations, biological interactions, and geological transitions. It has encountered changing oceans, shifting food webs, and repeated planetary disturbances. Through all of it, the lineage continued. The scale of that persistence is difficult to grasp directly. Approximately 80 million years is roughly 16,000 times longer than the entirety of recorded human history. It is approximately 4,000 times longer than the age of the oldest known civilizations. The duration extends so far beyond ordinary experience that intuition struggles to contain it. Human memory provides no useful reference point. Every language ever spoken, every city ever built, every scientific discovery, every work of art, every conflict, every migration, every empire, every philosophy, and every technological achievement occupies only a tiny fraction of the period through which this lineage has existed. Viewed another way, nearly everything that humans recognize as history fits within approximately 0.006% of the frilled shark's evolutionary timeline. The comparison is not intended to diminish humanity. It simply restores proportion. Human civilization feels immense because it contains the entirety of human experience. Against the backdrop of deep biological time, however, it occupies a remarkably brief interval. The frilled shark helps reveal that scale by serving as a living reference point, a lineage whose continuity stretches across durations that exceed ordinary comprehension. Its success also challenges certain assumptions about what survival requires. The surface world often rewards speed. It rewards flexibility, expansion, competition, and rapid response to changing conditions. Organisms inhabiting those environments frequently participate in evolutionary contests where the terms of survival shift continuously. The deeper ocean offered a different arrangement. There, patience could be rewarded. Precision could be rewarded. Stability itself could become an advantage. A species that matched its environment exceptionally well might continue doing so for extraordinary lengths of time without radical modification. In that sense, the frilled shark's history resembles a strategy, though no intention guided it and no intelligence designed it. Find an environment stable enough that being exactly what you are remains the correct answer. Remain within that environment. Allow time to pass. The result is not conquest. It is not domination. It is not victory over nature. It is a form of alignment so complete that generation after generation encounters essentially the same question and continues providing the same successful response. The frilled shark did not survive by forcing the world to accommodate it. It survived because it fit the world around it so well that the world rarely asked anything different in return. At this very moment, while conversations unfold in cities, while aircraft cross continents, while waves move across distant coastlines and clouds drift above oceans, a frilled shark is alive somewhere beneath the sea. Not in the past, not in a fossil bed, not in a museum drawer. Alive. Perhaps it is moving through water between 500 meters, which is about 311 miles, and 1,000 meters, which is about 621 miles, below a surface that may be calm, storm-covered, or illuminated by moonlight. Around it, the temperature remains near 4 degrees Celsius, or 39 degrees Fahrenheit. The pressure surrounding its body is immense, far beyond anything a human being could endure without protection. Yet, for the shark, these conditions are ordinary. They are home. Perhaps it is suspended almost motionless within the water column, supported by the buoyancy of its enormous oil-rich liver. Around it stretches darkness, uninterrupted by sunrise or sunset. No season announces itself. No weather system reaches this depth with meaningful force. Within that darkness, sensory systems continue their work. Tiny electrical signals ripple outward from living organisms hidden beyond sight. Specialized receptors detect those signals. Subtle disturbances pass through the water. Pressure waves travel outward from distant movements. Information arrives continuously, translated into a map of the surrounding environment without the need for light. Or perhaps the shark is already in motion. Its body moves in slow undulations through cold water, not hurried, not wasteful. Each movement reflects a design refined across immense spans of evolutionary time. Somewhere ahead, a squid changes direction. Muscles contract, water shifts, signals spread through the surrounding darkness. Distance narrows. The encounter unfolds as countless others have unfolded before it. The shark has no awareness of the role it occupies within human imagination. It does not know that people have devoted careers to studying its anatomy. It does not know that researchers have assembled fragments of evidence into tentative explanations. It does not know that images of captured individuals have traveled across the world through networks of communication unimaginable to the creatures inhabiting the deep. It has no relationship with any of those things. The surface world exists beyond its experience. For approximately 140 years, humans have known that the species exists. For approximately 80 million years, the lineage existed without that recognition. The difference between those durations reveals how recently the animal entered human awareness and how little that awareness has altered its life. Down below, the shark continues according to the same requirements that shaped its ancestors. Food must be found. Energy must be conserved. Offspring must eventually be produced. Generation follows generation. Meanwhile, above the ocean, the world transformed repeatedly. Landscapes changed. Civilizations appeared and disappeared. Languages emerged and vanished. Technologies accelerated. Knowledge expanded. Entire eras passed. The shark remained, not because it resisted change, but because the environment surrounding it never demanded much of it. The deep ocean continued offering the same fundamental conditions, and the organism occupying those conditions continued providing the same successful response. There is no final lesson waiting inside that observation. No moral conclusion emerges from it. There is only the fact itself. Somewhere beyond sight, beyond routine observation, beyond nearly everything that occupies human attention, an animal carrying tens of millions of years of history continues moving through water that most people will never see. Its existence does not depend upon recognition. Its survival does not require understanding. It simply continues. And while it continues, the larger truth remains with it. The living world is not limited to the portion humanity has explored. Vast regions still operate beyond direct observation. Entire stories remain unwritten. Entire lives unfold, unnoticed. Far below the surface, in cold water and enduring darkness, one of those stories is still being lived, exactly as it has been for ages beyond memory.